So welcome to the kanski lecture series, and in this part we are going to study about the hereditary fundus dystrophies chapter 15. And in this part we are going to discuss about the fundus dystrophies that are usually hereditary. So let's get started. Introduction. Journal. The hereditary fundus dystrophies are a group of disorders that commonly exert their major effect on the retinal pigment epithelium, RPE, photoreceptor complex, and the choriocapillaries to cause a range of a visual impairment. So mostly it affects the RPE, it affects the photoreceptors, and it affects the choriocapillaries. The most common example is retinitis pigmentosa. Hereditary retinal diseases are now the leading cause of blindness certification in the working age population age 16 to 64 years in the UK. So basically now it's the most leading cause of blindness in the working age population in the UK, which is between the age 16 to 64. Some dystrophies manifest in early childhood while others do not present until later in life. Isolated dystrophies have features confined to eye, while syndromic dystrophies are a part of a wider disease process that also affect the tissue elsewhere in the body. So sometimes there are isolated dystrophies which only affect the eyes. Sometimes there is an syndromic dystrophies which affect the other part of the body as well. In a patient with a retinal dystrophy, genetic sequencing should be undertaken to determine the exact causative gene, as mutation in a number of genes can cause the same clinical phenotype. Genetic heterogeneity and different phenotypes can be caused by the same genes, phenotypical heterogeneity. So basically there is a thing called genetic heterogeneity and the phenotypic. So sometimes there are different presentation of the same gene mutation, which is known as a phenotypical heterogeneity, and sometimes different genes causes the same disease, that is known as a genetic heterogeneity. Mutation in more than 250 genes have been linked to inherited retinal dystrophies, and a table of genes associated with retinal dystrophies can be found online. Identification of the causative gene allows the identification of the patient who might benefit from the clinical trials of a gene therapy. Sometimes some gene therapy also affect the some kind of gene mutations. So it is sometimes helpful to identify the cause of the gene which is causing this mutation. The mutation in different genes, genetic heterogeneity, can cause the same clinical phenotype in an individual with a retinal dystrophy. So we can have anatomy, inheritance. So first there is an anatomy. There are two types of a retinal photoreceptors. So after the RPE there is a photoreceptor layer, and we know that there are two types of photoreceptor. One is rods, another is cones. The rods are the most numerous, 120 million, and are of a densest concentration in the mid-peripheral retina. They are more sensitive in illumination and are responsible for night motion sense and peripheral vision. So almost 120 million rods are being present in the retina, and they are more dense at the mid-periphery, and they are for the most sensitive in dim illumination and are responsible for the night vision, motion sense, and peripheral vision. If rod dysfunction occurs earlier or is more severe than cone dysfunction, it will result in poor night vision, which is known as nyctalopia, and peripheral field loss, the former usually occurring first. Then there is the cones, which are far fewer in number, 6 million, and are concentrated at the fovea, so they are more dense at the fovea. They are more sensitive in bright light and mediate day vision, color vision, and central and fine vision. So it is opposite to the rod. It will mediate the day vision, color vision, and the central or fine vision. Cone dysfunction therefore result in poor central vision, impairment of color vision, dyschromatoxia, and occasionally problem with the day vision, which is known as a hemerotopia. So what is the inheritance of the dystrophies? Most dystrophies are inherited, but sometimes a new mutation, allelic variant, can occur in an individual and can subsequently be passed to the future generation. So inheritance can be autosomal dominant, recessive, X-linked dominant, mitochondrial, and digenic. So autosomal dominant dystrophies often exhibit variable expressivity and tends to have a later onset and milder course than a recessive disorder. So autosomal dominant have usually present later in life and are usually milder. Recessive dystrophies may be autosomal recessive or X-linked recessive. They usually have an earlier onset and are more severe course than autosomal dominant. In some cases, female carriers of X-linked recessive condition shows characteristic fundus finding. So recessive dystrophies are usually more severe and present earlier in life, and female carriers sometimes show characteristic fundus findings in X-linked conditions. X-linked dominant conditions are very rare and are usually lethal in boys, for example, Aicardi syndrome. Then there is a mitochondrial DNA. Mitochondrial DNA is inherited solely via a maternal line. Retinal dystrophies associated with mitochondrial DNA variants are extremely rare and occur as a part of a wider systemic disease. A maternal carrier will usually possesses a mixture of mitochondria, only some of which contain the dysfunctional gene, and the presence and severity of the resultant dystrophy in offspring depends on the proportion of a faulty mitochondrial inherited. So because the mitochondria is being inherited by the mother, so that is why the mitochondrial DNA a condition causes the more problem in the male. And most of the time, that the most common mitochondrial dystrophies are uh, Kern-Chiari syndrome, and another one which is very important for the case of the optic disease is the Leber hereditary optic neuropathy. Diagenic conditions are due to combined effect of mutation in two different gene. Mutation in the same gene can cause different phenotypical heterogeneity in an individual with a retinal dystrophy. So classification as well as division by the inheritance pattern dystrophies can be considered as generalized, in which the clinical effect involves the entire fundus, rod and cone or cone rod depending on which photoreceptor type is predominantly dysfunctional, or central local macula in which only the macula is affected. They can also be classified according to the element that is focus of the pathological process, for example photoreceptor, RPE or choroid, and by whether they are stationary, non-progressive or progressive. So they can be divided into different groups, like if the rods are affected or cones are defective, so we can have rod cone rod dystrophy, and we can have central dystrophy. We can have depending on which area is the pathological process, photoreceptor, RP or choroid, or whether they are stationary or progressive. So what are the investigations that we can do in a case of retinal dystrophy? Electroretinography is very important, which is also known as ERG. The electroretinography, ERG, means the retinal electrical activity. When stimulated by the light of adequate intensity ionic flow, principally sodium and potassium is induced in and out of the cell, such as the third potential is generated. The recording is made between the active electrode, either in contact with the cornea, or a skin electrode placed just below the lower eyelid margin, and a reference electrode on the forehead. The potential between the two electrodes is then amplified and displayed. The normal ERG is predominantly biphasic. So basically what we do is we place the one rod in front of the cornea, or we placed it on the skin below the lower eyelid, and another electrode is being placed at the forehead, which is basically the reference electrode. And when the eye is being stimulated by the light intensity, so there is a certain kind of potential generated, and the potential of the between the two electrons is then amplified and displayed, and this is how the ERG is being produced. So let's see figure 15.1. It is showing us there is an electrode placed in front of the cornea, and there is a reference electrode, and then the potential between them is generated, is being amplified and recorded. So this displayed a graph. So there are waves which is known as A wave and B wave. The A wave is a negative wave, whereas a B wave is a positive wave. A wave is being generated by RPE, and B wave is being generated by the bipolar cells. The A wave is an initial fast corneal negative deflection, generally by the photoreceptor, so it is a negative wave. B wave is subsequently slower positive large amplitude deflection. Although it is generated from the molar and bipolar cells, it is directly dependent on the functional photoreceptor, and its magnitude makes it a convenient measure of a photoreceptor integrity. So it can be indirect measure of an photoreceptor function as well. Its amplitude is measured from A wave trough to the B wave peak. It consists of a B1 and B2 subcomponents. and the former probably represent both rod and cone activity, and the later may represent mainly the cone activity, and it is possible to distinguish rod and cone responses with appropriate changes. The B wave is enhanced with the dark adaptation and increased light stimulus. So basically, B wave is being measured, amplitude is measured from the trough of the A wave to the crest of the B wave, and basically it can be divided into B1 and B2 component. B1 mostly represent the both rods and cone, and B2 mostly represent the cone activity. And B wave is being enhanced with the dark adaptation and increased light stimulus. Then there is a C wave, which is a third negative reflection generated by RPE and the photoreceptors. So the third wave is being generated by C waves is being generated by the RPE. The latency is the interval to the commencement of the A wave after the stimulus is being applied. So this is how, if the stimulus is applied here and it is a commencement of the A wave, it is known as a latent period. Implicit time is the interval from the stimulus to the B wave peak. So from the time of stimulus to the B wave peak is known as an implicit time. Electroretinography is used for the diagnosis of a range of different retinal disorders on the basis of a characteristic pattern of changes, as well as monitoring of disease progress in dystrophies and other conditions such as form of uveitis, for example birdshot retinochoroiditis, and drug toxicity, for example hydroxychloroquine. So this is a normal electroretinographic recording. So we will discuss it in the full field ERG. A standard full field ERG consists of 5 recordings taken during a diffuse stimulation of an entire retinal area and is used to assess the generalized retinal disorder but may not detect localized pathology. The first 3 are elicited after 30 minutes of dark adaptation scotopic, and last 2 after 10 minutes of the adaptation to a moderately brightly diffuse illumination photopic. It may be difficult to dark adapt children for 30 minutes, and therefore dim light mesopic condition can be utilized to evoke the predominantly rod mediated response to a low intensity white or blue light stimuli. So most of the time what we do is we take a full field ERG. First we adapt the patient to the dark for 30 minutes, and then we take a 3 reading, which is known as a scotopic readings. Then after 10 minutes of the moderately bright diffuse illumination, we take the 2 readings, which is known as photopic readings. But because the children are difficult to adapt for 30 minutes, so that is why we use the dim light conditions to evoke the moderate predominantly rod mediated response to white or blue light stimulus. So first we will discuss about the scotopic ERG, which consists of the 3 readings. Rod responses are elicited with a very dim flash of white or blue light, resulting in a large B and wave and a small or a non-recordable A wave. So after the dark adaptation, we rod response are elicited by dim flash white light or blue light, and it will result in a large B wave and a very small or a non-recordable A wave. So you can see this is a rod response. There is a very large B wave but a dim or non-recordable A wave. Combined rod and cone responses are elicited with a very bright flashlight, white flash, resulting in a prominent A wave and B wave. So second is basically combined responses, which is after dark adaptation we give a very bright white stimulus, and it results in A wave as well as a B wave as well. Then there is an oscillatory potential oscillated by using a bright flash and changing the recording parameters. The oscillatory wavelets occur on the ascending limb of the B wave and is generated by cells in the inner retina. So then we continuously stimulate the retina with the bright flashlight and record the parameters, and it results in an oscillatory pattern. Then there is a Photopic ERG cone responses are elicited with a single bright flash, resulting in an A and B wave with subsequent small oscillation. So, after the light adaptation, we stimulate the retina by the bright flashlight, which result in A wave and a B wave, and then there is a small oscillation. So you can see this figure here. So there is an A wave and B wave, and then there is a small oscillation. Then there is a cone flicker. So cone flicker is used to isolate cone by using a flickering stimulus at a frequency of a 30 hertz to which rod cannot respond. So then we use a flickering light stimulus which have a frequency of a 30 hertz to which the rods cannot respond. It produces a measure of amplitude and implicit time of a cone B wave. So what it will measure is basically the B wave amplitude and the implicit time. Cone responses are be elicited in normal eyes up to 50 hertz, after which point individual responses are no longer recordable. Critical flicker fusion. So most of the time 50 hertz can be used, flickering point can be used to elicit the cone responses, but after that it does not record. It is known as a critical flicker fusion. So this is how the cone flicker is being demonstrated on the ERG. So you can see that there are multiple B waves. Then there is a multifocal ERG. Multifocal ERG is a method of producing topographical map of a retinal function. The stimulus is scaled for variation in the photoreceptor density across the retina. At the fovea, where the density of the receptor is high, a lesser stimulus is employed than the periphery, where receptor density is low. As with conventional ERG, many types of measurement can be made. Both the amplitude and the timing of the trough and peak can be measured and reported, and the information can be summarized in the form of a three-dimensional plot, which can resemble a hill of vision. The technique can be used for almost any disorder that affects the retinal function. So this is a multifocal ERG. So you can see that basically it is giving us a three-dimensional map of the retina, in which you can see that the fovea is being stimulated with the much less intensity, whereas periphery is with the higher intensity. So you can see that here you can see that it is shown in the topographical map. Next is a focal ERG. Focal foveal ERG is used to assess the macular disease. Pattern ERG, a similar stimulus to that used in visual evoked potential. Pattern reversal is used to target ganglion cell function, typically in order to detect the subtle optic neuropathy. So in this ERG, what we use is basically the pattern stimulus. We have a certain pattern, and then we use as a stimulus to stimulate the retina, and it basically target the ganglion cell function to detect the subtle optic neuropathy. Then there is an electrooculography, which is EOG. So basically it is for the diagnosis of the visual pathway, if the visual pathway is intact or have some problem. The electrooculogram measures the standing potential between the electrically positive cornea and the electrically negative back of the eye. It reflects the activity of the RPE and the photoreceptor. This means that an eye that is blind as a consequence of a disease proximal to the photoreceptor will have a normal EOG. In general, diffuse or widespread disease of the RPE significantly affect the response. As there is a variation in EOG amplitude in normal subject, the result is calculated by the dividing the maximal height of potential in the light peak by the minimal height of the potential in dark trough. That is expressed as a ratio, which is known as Erdőn ratio, or as a percentage. The normal value is greater than 1.5 or 185%. So basically it measures the potential of the photoreceptor. If the photoreceptor is being if the disease is proximal to the photoreceptor, means that RPE or photoreceptor is being damaged, then is being affected. So that means any disease that is basically is proximal to that of RPE is being affected, and the photoreceptor is being affected. So basically what we do is basically we measure the potential in light potential and dark potential, and these are given is usually in a percentage as 185%, and the normal value is greater than 185%. So you can see that this is a principle of electrooculogram. So this is a positively charged cornea and negatively charged back of the eye, and both of them we can do that. There is a light peak crest and a dark trough that is being measured, and it is usually 185%, which is known as a Erdon ratio. Then there is a dark adaptometry. The dark adaptation is a phenomena by which the visual system adapts to the decreased illumination, and evaluation of this is particularly useful in the investigation of the nyctalopia. So what we do in the case of a dark adaptation is we adapt the patient to the dark and see if there is any kind of a night blindness problem. The retina is exposed to an intense light for a sufficient time to bleach 25% or more of rhodopsin in the retina. Following this, normal rods are insensitive to light and cone respond only to a very bright stimuli. So what we do is basically we expose the retina to a very bright light, and what happens is this bright light will bleach 25% or more of rhodopsin in the retina, and after that the rods become insensitive to light and cone responds only to a very bright light. Then we measure how much time it takes to recover from this bleaching. Subsequent recovery of the light sensitivity can be monitored by placing the subject in dark and periodically presenting the spot of light of varying intensity in the visual field, and ask the subject if they are perceived. So what we do is then we after stimulating the retina with the intense light, we then take the patient to dark, and we project the spots of light in different visual field and ask the patient if they can perceive or not. The threshold at which the subject just perceived the light is recorded. The flashes repeated at regular interval and the increased sensitivity of light of the eye to the light plotted the sensitivity curve. So 15.6 show us the sensitivity curve. So you can see here is how the rods and cones are being recovered. So you have placed the patient in dark, and you measure when the patient started responding to the light stimulus, and you can see how the rods are being recovered. So cone are being recovered at almost about 10 minutes, and rods being recovered almost after the 30 minutes. The cone branch of the curve represents the initial 5 to 10 minutes of darkness during which the cone sensitivity rapidly improved. The rod photoreceptor are also recovering, but more slowly during this time. So this is a cone branch which shows us that the cones are being recovered, but rods are recovering more slowly. The rod cone break normally occur after about 7 to 10 minutes when the cone achieve their maximum sensitivity, and the rods become perceptibly more sensitive than rod. So after 10 minutes the rods started becoming more sensitive. The rod branch of the curve is slower and represent the continuation of the improvement of rod sensitivity. After 15 to 30 minutes, the fully dark adapted rods allow the subject to perceive a spot of light over 100 times dimmer than would be possible with cones alone. If the flashes are focused on the fovea where the rods are absent, only a rapid segment corresponding to cone adaptation is recorded. So then next is a genetic testing which can be used to detect the retinal dystrophy. This should be undertaken in association with the geneticist who has access to laboratory that can undertake the necessary tests. Single gene testing is used in patients with family history of a known genetic mutation in order to confirm the molecular diagnosis. Sequencing using targeted panels of all known retinal dystrophy gene has been developed and yield high diagnostic rates. Once a genetic defect is identified, Sanger single gene sequencing can be undertaken to confirm the mutation. A monogenetic retinal dystrophy can develop at any age, and the genetic abnormality can usually be determined using the modern technique. So we can also do genetic testing to detect any kind of gene abnormality in cases of the retinal dystrophy. So that's it for the part 1. See you in the next lecture.